Hey, I'm Liver, the body's energy bank. I've got triacylglycerol saved up just in case. Ooh, I guess it's time. Time to send out some fatty acids.
Got him! I'm mitochondria, the powerhouse. Let's run beta oxidation and turn this fatty acid into pure energy.
As we have learned from our previous class here in biochemistry and even in physiology, our body undergoes a metabolic process known as cellular respiration to generate ATP, serving as the energy currency of the cell to carry out its normal function. Our knowledge and understanding of cellular respiration primarily focus on the conversion of chemical energy stored in foods, usually in the form of sugar or carbohydrates called glucose, into a usable energy in the form of ATP. However, in cases where there is little to no glucose available, how does the body generate ATP? That's what we are going to find out and explore in this session. Hello there, everyone. It's another day to learn and gain insights to apparently a newly introduced body mechanism for all of us. Our discussion will dwell on two major processes: beta oxidation and lipogenesis. Join us in turning a new page, a new phase, a new pathway towards our ATP quest. This discussion will focus on one of the major biological molecules we call lipids, particularly the fatty acids. Now, let's begin with beta oxidation. And a quick recap about fatty acids. Fatty acid contains a long hydrocarbon chain and a terminal carboxylate group. So if you may recall, fatty acid is a long chain consisting of carbons and hydrogens. And on its terminal end, there is what we call a carboxyl group or the COOH, the C double bond O, and another bond for OH. The hydrocarbon chain may be saturated, meaning there is no double bond or all bonds here are single, making them in a solid form. They can also be unsaturated, which contains at least one or more double bonds, making them in a liquid form. Fatty acid oxidation is the process where energy is produced by degradation of fatty acids. So this takes place when glucose level is very low. Basically, from the word oxidation, fatty acids will lose hydrogen atoms during the process. Remember the mnemonic oil rig? When we say oxidation, it is the loss of hydrogen. Reduction is gain of hydrogen. Now, let's go over with the types of fatty acid oxidation. Primarily, there are three types. First is beta oxidation, alpha oxidation, and omega oxidation. So in our discussion, we will just focus on beta oxidation. But to give you an overview, alpha oxidation predominantly takes place in the organs such as the brain or liver. In this process, one carbon is lost in the form of CO2 per cycle. So this occurs in the peroxisomes of our cell, wherein fatty acids in this process will undergo oxidation because of the presence of branch chain in the fatty acid, wherein there is a presence of particularly the methyl group. Whereas the omega oxidation, this is a minor mechanism but becomes important in conditions of impaired beta oxidation. This occurs in the endoplasmic reticulum of our cells, wherein fatty acid is converted or oxidized into dicarboxylic acid and will then be metabolized as the starting of that process. Okay, another distinction of the three types of fatty acid oxidation is the type of carbon that is being oxidized first in the process. So I have here stearic acid. So as you may recall, this is the carboxyl group. So meaning the carbon in the carboxyl group will be tagged or will be named as the carbon 1. The carbon next to the carbon 1 or the carbon in the carboxylic group, the carbon 2, is what we call as alpha carbon. So this is the first carbon that is being oxidized in alpha oxidation. Whereas the next carbon or the carbon next to the alpha carbon, the carbon 3, is what we call as the beta carbon, which is being oxidized first during beta oxidation. And lastly, omega carbon is the carbon that is on the opposite end of the carboxyl group. So since this is our carboxyl group, so the carbon here is what we call as the omega carbon, specifically omega 1. This is omega 2, and this is omega 3, and so on. Now let's focus on beta oxidation. So beta oxidation is a process that is responsible for breaking fatty acids to generate energy. Unlike cellular respiration, wherein it is the breaking of glucose to generate energy, beta oxidation will break fatty acids instead to generate energy. And the same with cellular respiration, it occurs in the mitochondria, and beta oxidation will result in the sequential removal of a two-carbon fragment, acetyl-CoA. After the production of acetyl-CoA, it is fed directly into Krebs cycle, and oxidative phosphorylation will follow afterwards. So beta oxidation occurs in liver, kidney, and heart, but it does not occur in the brain, mainly of two reasons. First, there is limited enzymatic activity in the brain that will help carry out beta oxidation, meaning beta oxidation, just like cellular respiration, dagan og enzymes involved, but our brain has limited enzymatic activity sa kanina mga enzymes, making it na beta oxidation will not occur. And second, most of the fatty acids in the brain are branch chain. So there is the presence of branch chain groups such as phytanic acid. Phytanic acid has methyl groups. So beta oxidation cannot occur. Instead, sa kung giingon ganina katong alpha oxidation ang mo occur sa brain. And lastly, beta oxidation is strictly aerobic, meaning sa beta oxidation, there is a need of oxygen. Nga naman, as you may recall sa oxidative phosphorylation dito sa electron transport chain, no? And chemiosmosis. So, ang oxygen will serve as the final electron acceptor. So, if walay oxygen, then beta oxidation cannot serve its purpose nga mo generate of energy using the fatty acids in replacement sa glucose when there is little to no glucose available. So, if walay oxygen, dili maka-proceed sa or the ATC, the electron transport chain will stop. So, since ang beta oxidation is nag-agad man po siya after sa Krebs cycle kay mosulod man siya sa electron transport chain, if walay oxygen dito, mohunong ang electron transport chain, and then walay man-generate na energy, so beta oxidation will also stop or will not serve its purpose. So prior to beta oxidation process proper, there is first the need to mobilize and activate the fatty acids. So triacylglycerols are highly concentrated sources of metabolic energy due to their reduced and anhydrous nature. They consist of three fatty acids esterified to a single glycerol backbone and can yield over twice the energy of carbohydrates or proteins. So if kung inyo itong nahinumduman, ang katong choco-choco na to na nagdrota, diba, there is the single glycerol backbone and then naay tulo ka fatty acids nga nasumpay dito through the process of esterification. And it is said that triacylglycerols can yield over twice the energy of carbohydrates or proteins. And let us discover if this is true ba gyud during nasa calculation sa to ang ATP on the calculation part. Triacylglycerols have lower toxicity than free fatty acids, allowing them to accumulate at higher concentrations. So, before beta oxidation, there is a need na fatty acid will be mobilized from triacylglycerols through the process like lipolysis or lipophagy. releasing free fatty acids for degradation in the mitochondria. So, dapat, ang mosulod sa to ang beta oxidation process kay ang katong free fatty acid, meaning dili ang katong triacyl glycerol, dili ang katong naayang glycerol na backbone niya, tulo ka buok fatty acid. Dapat lang yun. Ang kato lang, isa lang siya ka free fatty acid. We also have what we call fed state and fasted state. So in fed state, excess energy leads to the storage of fatty acids as triacyl glycerols within adipose tissue. So unsay pasabot sa fed state? From the word fed, meaning gikaon. And sa tuang gikaon, there is carbohydrates, proteins, lipids, and fats. Since naamantay gikaon or nag-consume man tag foods that are rich in carbohydrates, so naatay source of glucose, which will then be used para sa pag-generate sa ATP through cellular respiration. So, ang ato ang gikaon ng mga fatty acids or ang fats, ang lipids, dili siya magamit for beta oxidation kaya naamay glucose. So, asa siya padulong? So, this will be stored within the adipose tissue as triacyl glycerols. And during fasted state, so sa fasted state, as this is prominent sa nag-diet or during fasting, glucose availability is reduced kay ang kuan ani is ang pagkaon is reduced or limited. So ang source sa glucose will also be reduced. So meaning our body cannot perform cellular respiration and will resort adto sa beta oxidation. So these stored lipids, o mga na-stored na lipids sa atuang adipose tissue will be mobilized to meet the body's energy demands. So the fatty acids will be broken down to generate energy. And for as an overview for the steps in beta oxidation of fatty acid, first is the activation of fatty acids occurring in the cytosol. Second is the transport of fatty acids into mitochondria. And lastly is the beta oxidation proper in the mitochondria as well, but specifically in the mitochondrial matrix. Let's go first with the even chain fatty acid oxidation. So when we say even, we are talking here about the number of carbons that are even. So we have 10, 12, 14, 16, 18, and so on. And we will also tackle about the odd chain fatty acid oxidation, which is those number of carbons that are in odd. So like 11, 13, 15, 17, and so on. Now, let's go to the steps in beta oxidation of fatty acid. So again, there is a need first to activate the fatty acids in the cytosol. Let me show the reaction first. So we have here a fatty acid. So as you can see sa kanina diagram, ATP is a requirement for this process. So ATP will be hydrolyzed using the enzyme or with the enzyme thiokinase into pyrophosphate and AMP. So again, ATP is adenosine triphosphate. So meaning there are three phosphate bonds. PPI, pyrophosphate, is consists of two phosphate bonds. So meaning from three phosphate bond, na-hydrolyze siya PPI or pyrophosphate nga two phosphate bonds. And we have also here ang iyang isa pang hydrolyze is AMP or adenosine monophosphate. So isa lang ka phosphate bond. So aari sa tao na sa pyrophosphate. So pyrophosphate will be further hydrolyzed into two inorganic phosphate by the use or by the help of the enzyme pyrophosphatase. So nanatay duha ka inorganic phosphate, so ensuring that there will be efficient oxidation. So ang katong adenosine monophosphate ang isa sa na-hydrolyze atong ATP will join the fatty acid. So diri siya mo join So we have C double band O sa atong carboxyl group, nawala ang OH, mailisan siya og AMP, and this is called as acyl adenylate. And then acyl adenylate will be further hydrolyzed, wherein we have here a CoH. So this is just coenzyme A with a thiol group. So thiol group is SH, sulfhydryl group. So with CoH, ang kaning acyl adenylate, mawala ang AMP. So ang AMP diri, once na ang CoH, mawala ang AMP, and then CoA will replace. So from acyl adenylate, it will now be acyl CoA, so the fatty acid, and then joining CoA or coenzyme A, forming acyl CoA. So it is important to note, okay, magamit ni siya during sa katong calculation na sa para sa net ATP nga generated na. Although there is only one ATP nga directly used in the process, but the hydrolysis of the pyrophosphate into two inorganic phosphate uses energy of two high energy bonds. So, two high energy bonds ang gi-use para ma-hydrolyze ang pyrophosphate into two inorganic phosphate. So, it appears nga there are two ATP molecules nga na-consume during sa activation process sa fatty acid. Although, ang atong nakita diri is only one. Again, I will repeat. Although ang naa diri is only one ATP ang directly nga nagamit sa process, but during the hydrolyzation of the pyrophosphate to two inorganic phosphate, two high energy bonds ang nagamit para makaproceed ani nga process. So therefore, it is equivalent to two ATP molecules ang na-consume during sa activation. So, and also to repeat again, so from fatty acid, it will now be acyl adenylate, and then the addition of CoA uh, results to the repulsion of AMP, so mawala ang AMP, there is ato ang acenyl adenylate, acyl adenylate, and will then be formed or will then produce acyl CoA kanina sa ubos. Proceeding to the next step, which is the transport of the fatty acyl CoA from the cytosol to the inner mitochondrial membrane. Please take note that fatty acyl CoA is impermeable to the inner mitochondrial membrane, so dili siya kaagi. Thus, a carnitine shuttle will facilitate this process. So carnitine acyl transferase 1 transfers the acyl group from CoA to carnitine, forming acyl carnitine. This is then transported across the inner membrane. Inside the mitochondrial matrix, carnitine acyl transferase 2 transfers the acyl group back to CoA, regenerating fatty acyl CoA for beta oxidation and releasing carnitine. This cycle repeats. Referring to this figure, so again, we have here the cytosol, the outer mitochondrial membrane, the intermembrane space, the inner mitochondrial membrane, and the mitochondrial matrix. So from the cytosol, the acyl CoA will be transported into the mitochondria. However, it is impermeable, so meaning di siya kadali-dali o pass through sa mitochondrial membrane. So para makasulod siya sa mitochondria, acyl CoA will undergo carnitine shuttle transport, wherein ang acyl CoA will uh, pass ari sa carnitine acyl transferase 1. So ang CoA or ang coenzyme A sa acyl CoA kay mawala, and it will be replaced with carnitine. So iniagi niya diri, acyl CoA will be acyl carnitine. And then it will pass through the transporter sa inner mitochondrial membrane. And afterwards, moari dayon siya sa carnitine acyl transferase 2 para ang atong acyl carnitine kay mabalik into acyl-CoA. So again, pambalik ko na. Acyl-CoA diri sa cytosol. Moagi siya diri sa carnitine acyl transferase 1. CoA will be replaced with carnitine. So acyl carnitine will then pass through the transporter sa inner mitochondrial membrane, and then from here will mudritso siya sa carnitine acyl transferase 2, wherein acyl carnitine will be uh, synthesized back to acyl CoA. So nakasulod na sa mitochondrial matrix ang acyl CoA para mo proceed sa beta oxidation proper. Let's proceed with the third. This is beta oxidation proper. So each cycle of beta oxidation, liberating a 2-carbon unit acetyl-CoA, occurs in a sequence of 4 reactions. First is the oxidation by FAD. Second is hydration. Third is oxidation by NAD. And fourth is cleavage. Let's have first with oxidation by FAD. So, nakasulod na ang atong acyl-CoA sa mitochondrial matrix. Beta oxidation proper will proceed sa mitochondrial matrix. So, acyl-CoA undergoes dehydrogenation by an FAD-dependent flavoenzyme acyl-CoA dehydrogenase. A double bond is formed between alpha and beta carbon. So, in this case, that's the 2 and 3 carbons, or carbon 2 and carbon 3. For here, so nanatay acyl-CoA. Again, yellow is the sulfur. This is the thiol group of the OA. And then we have here the carbon 1 with the double bond O. We have alpha carbon, the beta carbon, and the R group. So, mo ni ang katong remaining group sa tuang fatty acid chain. So, how does the oxidation by FAD occurs? So, we have here FAD, flavin adenine dinucleotide. And then, we have here an enzyme acyl-CoA dehydrogenase. So, one hydrogen from alpha carbon and one hydrogen from the beta carbon will be dehydrogenated. So, meaning, ang separate siya sa ato ang acyl-CoA. And this two hydrogen will be added sa FAD with the help of the enzyme acyl-CoA dehydrogenase para mo-form ang FADH2. And then, this reaction will result to alpha-beta enoyl-CoA since na-dehydrogenate naman ang duha ka hydrogens dire, here and here. So, wala na hydrogen nga ni bond. So, this would result na ang alpha and beta carbon will be bonded with a double bond, forming alpha-beta enoyl-CoA. Now, let's proceed sa second. This is hydration. So, from the word, no, it entails with the addition of water. So, enoyl-CoA, or the alpha-beta enoyl-CoA, will be undergoing hydration of the double bond by the enzyme enoyl-CoA hydratase to form 3-hydroxyacyl-CoA. So, please refer here. So, we have here the alpha-beta enoyl-CoA from the product from the first process. And then, by the enzyme enoyl-CoA hydratase, so, and there is water, H2O, it would form 3-hydroxyacyl-CoA. So, inyong makita, again, akong balikon. So, mao ni siya ang ato ang una, mao ni ato ang reactant diri with addition of water, mao na ni siya. So, nibalik siya ang katong na dehydrogenate na duha ka hydrogen nabalik, nawala ang double bond diri, pero naay addition of oxygen diri sa to ang beta carbon. Meaning, mao na siyang gitawag siya og 3-hydroxyacyl-CoA kay 1, 2, 3 hydroxy. So, naay hydroxyl group na na-form and then acyl-CoA. Proceeding, the third is oxidation by NAD. 3-hydroxyacyl-CoA dehydrogenase catalyzes the second oxidation and generates NADH. The product formed is beta-keto-acyl-CoA. Refer to this one. So we have here the 3-hydroxy-acyl-CoA. We have NAD+. We have hydroxy-acyl-CoA dehydrogenase. So in the process, so this is oxidation by NAD. The hydrogen, the two hydrogens in the beta carbon will be dehydrogenated. And then one hydrogen will combine with NAD+, forming NADH. And then there is an excess of one hydrogen. So that's NADH plus H. And then this reaction is catalyzed by the enzyme hydroxy-acyl-CoA dehydrogenase. The reaction will result to the formation of beta-keto-acyl-CoA. So as you can see, ang ato ang beta carbon is double bond na siya with oxygen. So ang atong beta carbon is na ay carbonyl group, which is signification sa ketone. So as for the last stage of the beta oxidation proper, the final reaction in beta oxidation is the liberation of a 2-carbon fragment, acetyl-CoA from acyl-CoA. This occurs by a thiolytic cleavage catalyzed by beta-keto-acyl-CoA thiolase or simply thiolase. The new acyl-CoA containing 2 carbons less than the original re-enters the beta oxidation cycle. The process continues till the fatty acid is completely oxidized. So refer here. We have the beta-keto-acyl-CoA. We have an enzyme thiolase plus another CoA. The result would be acyl-CoA plus acetyl-CoA. So as you can see, so ang ato ang beta carbon na anay double bond oxygen plus the R group, then the sulfur and the CoA forming acyl-CoA. And then, ang kaning acetyl-CoA, mao ni siya ang katong carbon 1, the alpha carbon, and then the addition of the sulfur and the CoA para acetyl-CoA. And as you can see, ang atong acyl-CoA diri is less than 2 carbons na siya. Kay pagsugod na to, di ba? Napat to diri ang kanima ni nasumpay pa na siya diri. Mo to acyl-CoA na siya. Pero since after the series of processes, ang kani siya niri na porma naman og acetyl-CoA which is 2 carbon. So less than 2 carbon na lang ang acyl-CoA nga na form after diri. And mao na siya ang iyahang mechanism sa next once mo re-enter ni siya sa oxidation, kada mahuman ang cleavage kay mo less than na siya og 2 carbon kay ang kanang 2 ka carbon mao na siya ang mo-comprise sa atong acetyl-CoA. So ang acyl-CoA, as I have mentioned, mo re-enter siya sa beta oxidation process kay taas man niya atong fatty acid chain, taas ni siya, di ba? Nani siya depende lang sa kung pila ka carbons. So dapat man ang katong tanan carbons will be oxidized. Pero diri sa tong first pa lang na process or first pa lang na round is ang duha pa ka carbon ang na-oxidize into acetyl-CoA. So ang katong uban i-oxidize pud to into acetyl-CoA, and to para mahug nga murag siya ang mo restart sa process, need na to ang acyl-CoA. So ang acyl-CoA again mobalik siya mo re-enter sa oxidation cycle para ang katong ubang carbon will be oxidized until nga ang whole fatty acid will be oxidized. Meanwhile, ang kaning acetyl-CoA will be fed into the Krebs cycle. So, mo proceed na sa Krebs cycle and sa oxidative phosphorylation wherein na ay electron transport chain and chemiosmosis para ma-generate ang ATP with the NAD, the FAD, and the acetyl-CoA. So, ang pag-calculate or ang pag-compute sa kung pila ang na-generate nato nga ATP for a specific fatty acid will be tackled na later. And that ends sa whole na beta oxidation process. Okay, so ang pag-trace nato sa tong acetyl-CoA no nga mao'y ma-produce in each round or in each cycle sa pag-oxidize sa ato ang fatty acid chain, the even fatty acid chain. Mosulod siya sa Krebs cycle and it will follow the usual nga Krebs cycle sa atuang cellular respiration with acetyl coenzyme A as mao'y mo start sa process. So it would undergo a lot of synthesis of compounds pero dili na to siya tagsa-tagsaon kay bawo naman ta and na-discuss na pud siya. So ang ato lang focusan here again is to take note that in each molecule of acetyl-CoA na mo undergo sa Krebs cycle will result to the production of 3 NADH. So we have here 1, 2, and 3. There's also production of 1 PADH2, so kani diri nga part, PADH2. And we also have the production of 1 GTP. So, mao na siya equivalent to 1 ATP. So, para sa tong calculation later, para dali siya, we will just put that acetyl-CoA has an equivalent ATP of 10. Kaya nga naman, di ba? So, there are 3 NADH and NADH, 1 NADH is equivalent to 2.5 ATP. So, 2.5 times 3 is 7.5. And then we have FAD-H2. 1 FAD-H2 is equivalent to 1.5. So 7.5 plus 1.5 is equals to 9. And then we also have here 1 ATP in the form of GTP. That's 1. So 9 plus 1, that's 10. So later, calculate na to sa number of ATP na generates sa whole process. We will just ilahos na lang nato that acetyl-CoA will equals to 10 ATP or will be having an ATP equivalence of 10. Okay, so let's proceed to the odd chain fatty acid oxidation. So odd chain here is that the number of carbons in the chain is in odd number. So 11, 13, 15, 17, and so on. So just like sa even chain fatty acid oxidation, mo generate gyapon siya og acetyl-CoA. It's just that ang differences nila is sa last na round or sa last cycle, instead nga ang ma-generate niya is acetyl-CoA, ang ma-generate niya is propionyl-CoA. Again, please remember that acetyl-CoA is composed of 2 carbons. Propionyl-CoA is composed of 3 carbons since odd chain man gud ni siya. So meaning naay isa nga dili ma-partner. So wala siya pares. So dili siya makabuhat og acetyl-CoA. So instead, ang katong last 3 nga carbon sila ang magkuyog to form propionyl-CoA. So propionyl-CoA, we have here ang last na part sa chain. Okay? Okay. So next is the carboxylation of propionyl-CoA. So there will be an addition of the carboxyl group. So by the enzyme propionyl-CoA carboxylase. So as you can see, diba, na diri sa alpha carbon, no, hydrogen siya diri sa propionyl, pag abot diri sa carboxylation, na-attach ang carboxyl group. So nanatay carboxyl group ari. And also to progress this process, need ang ATP and then ma-synthesize siya onto ADP plus the PI or inorganic phosphate. And then biotin will act as a cofactor here. And then this reaction would result to dimethylmalonyl-CoA. After, ang dimethylmalonyl-CoA with the help of methylmalonyl-CoA epimerase will epimerize the dimethylmalonyl-CoA into L-methylmalonyl-CoA. So, ang pag-synthesize ani sa next na compound kay ang enzyme niya will act lang yun specifically sa iyang L na epimer. So, from D-methylmalonyl-CoA, mahimo siyang L-methylmalonyl-CoA. So, ang ato lang bantayan here is it's just that no ang CoA kay ni join dito sa katong gi-add na carboxyl group and then ang kaning another na to nga O diri Kay nibalhin ra sad diri sa another carboxyl group ang katong original na carboxyl group. And then, L-methylmalonyl-CoA will undergo isomerization by the help of methylmalonyl-CoA mutase. So L-methylmalonyl-CoA, then napodjay coenzyme na B12 or cobalamin. This is a crucial cofactor para mo-proceed ang process. And this will result to succinyl-CoA. So this is just an isomerization. So nabalira ang atong hydrogen diri, ang isa ka hydrogen diri sa third carbon, nabalhin dito sa second carbon, and then ang katong added na carboxyl group with the CoA na is nabalhin na diri sa tuang third carbon. So muna siyang succinyl CoA. So proceeding, kay mo enter na siya sa atoang citric acid cycle. So this is the process, the whole process. And then, ang katong succinyl-CoA nato will enter Krebs cycle. Pero as you can see, ang iyahan na lang ma-enter sa Krebs cycle is katunga na lang sa process sa usual na Krebs cycle. Kaya nga naman, wala siya nagsugod sa acetyl-CoA. Instead, gasugod siya sa succinyl-CoA. So as you can see, throughout the process, instead nga ang ato ang NADH kay tulo, sa kaning sa add chain, since sa succinyl-CoA man ta nagsugod, isa na lang ka NADH ang na-generate, naka-generate yapon og FADH2 and also 1 GTP. So i-add pod nato ni ang mga kani sila, i-add ni nato ang ilahang ATP equivalent sa katupong na generate sa pagpo-form sa mga acetyl-CoA. So we have here 2.5, 1.5 and 1. If you have any questions or any clarifications, feel free to ask because I will be addressing them in our face-to-face na nga session kay medyo so they explain ang problem set or ang computation pod in a video recording. So before I will proceed lang sa problem set, I will just kanang present to you a simplified version na sa kaning nga whole topic for you to understand as well sa pag-generate sa pila ka number of ATP per cycle, per round, and also ang pag-calculate na sa whole na ATP generated sa whole process. So that's all for beta oxidation. And after my discussion on the problem sets and the clarifications and all your questions, we will also proceed to the other half of our topic, which is lipogenesis. Thank you everyone for listening.
A pleasant day, everyone. I'm Engineering Blackfoot, and I'm here to talk about lipogenesis. Lipogenesis is a metabolic pathway that synthesizes fatty acids from acetyl-CoA, primarily occurring in the cytosol. It is crucial for energy storage, particularly in the fed state, when the body prioritizes storing energy rather than utilizing it. Unlike beta oxidation, which breaks down fatty acids, lipogenesis involves the construction of fatty acids, highlighting its distinct biochemical processes. Now let's take a look at the key differences between beta oxidation and lipogenesis. When it comes to the reaction sites, lipogenesis occurs in the cytosol, while beta oxidation takes place in the mitochondria. For the enzymes involved, we have fatty acid synthase and acetyl-CoA carboxylase for lipogenesis, whereas beta oxidation involves multiple independent enzymes. For the intermediate carriers, coenzyme A is used in beta oxidation, while acyl carrier protein is utilized in lipogenesis. As for the coenzymes, beta oxidation requires FAD and NAD+, while lipogenesis uses NADPH. As to the carbon atom handling, Beta oxidation removes two carbon atoms at a time, while lipogenesis adds two carbon atoms at a time. Let's get into the stages of lipogenesis. We can conceptually divide the pathway of fatty acid synthesis into three stages. A. Export of acetyl-CoA to the cytosol. B. Carboxylation of acetyl-CoA to malonyl-CoA. And C. Sequential addition of two carbon fragments to form palmitate. So here's the general overview of the three stages. All right, let's start with the export of acetyl-CoA to the cytosol. So these are what happens, but let's just take a look at the diagram. As you can see here in the diagram, we have here the cytosol and the matrix. Mitochondrial citrate is formed from the condensation of acetyl-CoA with oxaloacetate in the citrate synthase reaction, which we encountered previously as part of the Krebs cycle. As we already know, the outer mitochondrial matrix is freely permeable to acetyl-CoA, as well as many other substances, such as citrate, malate, and pyruvate. The inner mitochondrial membrane, however, is not permeable to acetyl-CoA. That's why the export of acetyl-CoA to the cytosol is achieved indirectly via the citrate shuttle. This one. Essentially, acetyl-CoA produced in the mitochondria is carried by citrate into the cytosol. A selective transporter in the mitochondrial inner membrane enables export of citrate to the cytosol. Cytosolic citrate is a substrate for citrate lyase, and the products are acetyl-CoA and oxaloacetate. This reaction involves ATP cleavage and is distinct from the mitochondrial citrate synthase reaction. And let's continue to the next part, which is from the cytosol to the matrix. Take a look at the diagram here. While acetyl-CoA is destined for fatty acid synthesis, oxaloacetate, the other citrate lyase product, continues through the citrate shuttle. First, a cytosolic enzyme of malate dehydrogenase catalyzes the conversion of oxaloacetate to malate using NADH. Next, malate is oxidatively decarboxylated to pyruvate, catalyzed by malic enzyme. The electrons from malate are captured as NADPH, a reducing agent used in the latter steps of fatty acid synthesis. Once pyruvate has been formed, it can re-enter the mitochondria and reform oxaloacetate to complete the cycle. Thus, as we have seen in the Krebs cycle, the citrate shuttle accepts acetyl-CoA as a two-carbon input. The Krebs cycle produces two net CO2 molecules per turn, whereas the citrate shuttle outputs two carbons per turn in the form of one cytosolic acetyl-CoA molecule. Let us now take a look at the next stage of lipogenesis, which is the carboxylation of acetyl-CoA to malonyl-CoA. The carboxylation of acetyl-CoA is a principal regulatory site of fatty acid biosynthesis. Once acetyl-CoA is converted to malonyl-CoA, it cannot easily revert back to its original form. The cofactor that helps facilitate the conversion is biotin. Alright, now let's take a look at the diagram shown here. So you can see here, the reactants are acetyl-CoA, HCO3 or bicarbonate, which acts as a source of carbon dioxide, and ATP, which provides the energy for the reaction. Bicarbonate from biotin is then donated to the methyl carbon of acetyl-CoA to form malonyl-CoA, at a separate site in the enzyme. So the products are malonyl CoA, ADP, and inorganic phosphate. This other diagram shows us a closer look on the biotin enzyme complex. Biotin, the cofactor, is first activated with ATP, leading to the formation of a biotin enzyme complex or E-biotin. The bicarbonate ion is utilized and carbon dioxide is released from the formation of malonyl CoA. The enzyme attaches the carbon from bicarbonate to the biotin and then to acetyl CoA, ultimately forming malonyl CoA. Let's go back here. Acetyl CoA carboxylase is controlled by both allosteric regulation and covalent modification or protein phosphorylation. The positive regulated citrate is a feed-forward activator of acetyl CoA carboxylase, similar to the activator of pyruvate kinase, fructose 1,6-P2. Negative modulator, fatty acyl CoA, decreases acetyl CoA carboxylase activity, reflecting an increase in fatty acids and hence less need for de novo biosynthesis. On this next slide, we are going to talk about the regulatory circuit of lipid metabolism. The triacylglycerol synthesis involves the conversion of fatty acyl CoA to triacylglycerol, storage form of fat. Fatty acyl CoA is a compound central to both fat synthesis and oxidation. Now, there are two competing pathways: fatty acid synthesis and fatty acid oxidation. In fatty acid oxidation, fatty acid CoA can enter the mitochondria through conversion to fatty acyl carnitine via carnitine acyltransferase 1. This one, CAT. Once inside the mitochondria, fatty acyl carnitine can be converted back to fatty acyl CoA for oxidation to generate energy. On the other hand, in fatty acid synthesis, acetyl CoA is a substrate for the synthesis of malonyl CoA. Malonyl CoA serves as a building block for fatty acid biosynthesis. So the citrate here activates fatty acid synthesis by increasing the levels of acetyl CoA. The hormone insulin promotes fatty acid synthesis and inhibits fatty acid oxidation, enhancing lipid storage. On the other hand, glucagon or epinephrine are hormones that trigger fatty acid oxidation and inhibits synthesis, promoting energy release from fats. AMPK is activated by AMP and it promotes fatty acid oxidation while inhibiting synthesis. The interplay between these components illustrates a feedback mechanism. High energy status leads to increased triacylglycerol synthesis, while low energy status, high AMP, leads to reduced synthesis and enhanced oxidation. Finally, let's discuss the third stage of lipogenesis, the sequential addition of two carbon fragments to form palmitate. In mammals, the ensuing reactions of fatty acid biosynthesis are catalyzed by the enzyme palmitate synthase. Like the pyruvate dehydrogenase complex of mitochondria, the individual reaction products remain attached to the enzyme complex. They are passed from one active site to the next until the final product, palmitate, is released. Structurally, palmitate synthase in eukaryotes is a dimer in which each strand contains all 7 active sites for the reactions of fatty acid synthesis. These reactions are catalyzed by physically separate individual enzymes in bacteria. Alright, so let's start with loading. This is ACP-SH, the thiol form of ACP or acyl carrier protein. It plays a crucial role in fatty acid metabolism by carrying acyl groups. So let's first start with malonyl CoA. You can see in the diagram that malonyl CoA is reacted with ACP-SH and CoA. With this, The malonyl group is transferred from malonyl CoA to ACP, forming malonyl ACP. This reaction creates a malonyl ACP molecule shown by the linkage between malonyl with carbonyl functionalities and ACP via thioester bond. Same thing happens with acetyl-CoA. It reacts similarly with ACPSH and CoA. The acetyl group is transferred from acetyl-CoA to ACP, forming acetyl-ACP, in which the acetyl group is linked to ACP via thioester bond. Lastly, the enzyme ESH reacts with acetyl-ACP, forming acetyl S synthase. Number 2, condensation. The carbonyl carbon from acetyl Se is attacked by the nucleophile, the malonyl component. And these red arrows here in the diagram indicate electron flow toward forming new bonds. Now, during the reaction, one molecule of carbon dioxide is released as a byproduct. This step typically involves the removal of a carboxyl group, which is characteristic of malonyl compounds. The thioESH is also regenerated during the reaction, indicating that the acyl carrier has been transferred. After the nucleophilic attack and decarboxylation, the product formed is acetyl ACP. This product contains two carbonyl functionalities and connects back to ACP, which is crucial in fatty acid biosynthesis. Third, we have the first reduction. This reduction reaction involves an acyl carrier protein and an ADPH. The reaction produces a keto group to reduces a keto group to a hydroxyl group. The acetyl ACP receives electron from NADPH, and a hydride ion from NADPH is transferred to this carbonyl atom, which changes its structure to an alcohol. Fourth, we have dehydration. As we already know, dehydration is a chemical reaction that involves the removal of water from a molecule, typically resulting in the formation of a more complex molecule. This process proceeds by the usual route of protonation of the hydroxyl group and loss of water coincident with electron migration to form the double bond. Double bond produced is in the trans configuration, similar to the fumarase reaction of the Krebs cycle. Finally, we have the fifth reaction, the second reduction. The double bond is reduced by hydride addition from the cofactor NADPH, thus NADPH is used twice in each round to convert the carbonyl group to a methylene group. The product is butyryl as ACP. In the chain elongation process, the sequence we just discussed is repeated in the next round, but with a slight variation. The full carbon butyryl as ACP is used in place of acetyl as ACP. The full carbon butyryl as ACP is used in place of acetyl as ACP. In subsequent cycles, increasingly larger acyl ACP is used. Thus, the T steps of fatty acid biosynthesis are yet another example of repetition variation, a similar way to fatty acid oxidation. The process of reloading malonyl and repeating the condensation, reduction, dehydration steps continues until the fatty acid chain reaches the desired length, in this case, 16 carbons for palmitate. Once palmitoyl CoA is formed, a thioesterase specific for 16 carbon chain catalyzes palmitoyl as ACP. Hydrolysis to form palmitate and ACPSH. Cells synthesize various fatty acids starting from palmitate using distinct enzymatic reactions, such as those for elongation and desaturation. These enzymes are embedded in the endoplasmic reticulum. They use acetyl-CoA for elongation and flavin-dependent non-energy linked oxidation to produce double bonds. Let us now discuss the triacylglycerol formation. Most of your lipids in the body are stored as triacylglycerols. The two hydroxyl groups of glycerol P exit fatty acyl chains through successive acylation reactions with fatty acyl CoA. Let's start with the conversion of glycerol phosphate from the hydroxyacetone phosphate, or DHAP. So the hydroxyacetone phosphate is reduced and transformed into glycerol phosphate by NADH and glycerol phosphate dehydrogenase. Next is the production of lysophosphatidic acid, in which the glycerol phosphate undergoes acylation with the help of the enzyme fatty acyl transferase. A fatty acyl CoA molecule is added, which leads to the formation of lysophosphatidic acid. Next is the formation of phosphatidic acid. This is the phosphatidic acid. So formation goes so lysophosphatidic acid is further acylated. Another fatty acyl transferase operates here to add an additional fatty acyl group from acyl CoA to form phosphatidic acid. Next up is the conversion of diacylglycerol. Here, the phosphatidic acid undergoes a dephosphorylation process by the enzyme phosphatidic acid phosphatase. This reaction yields diacylglycerol. Finally, the diacylglycerol is then acylated. Another fatty acyl transferase catalyzes this reaction, forming the final product of this series. Is the triacylglycerol or triacylglyceride, a molecule consisting of 3 fatty acid chains into a glycerol. So that's it for group 2. Here are our contributions. Thank you so much for listening, and hope you enjoy and hope you learned something. Bye.
Energy complete. You're welcome, buddy.
Good job, Mito. We make a great team.